Good morning, everyone. Can you see the slides?
Yes,
Good
sir.
morning, sir.
Okay, so I will start. So today we are going to discuss about lipoproteins. Basically, what are lipoproteins? What it is, structure, classification. Classification is important for you, and its metabolism and significance in the body, and how it is important for lipid metabolism and lipid transport. So basically, lipoproteins are basically the carriers of the lipids, whether absorbed from the diet or the lipid synthesized in lipid or liver or adipose tissue. So lipids absorbed from the diet and synthesized by liver and adipose tissue. So either lipid you can take from the diet or it can be synthesized in the body. Okay, so all the lipids have to be transported between various cells and organ for utilization and storage. Right, utilization we saw that while where all lipid is used, and it is stored as source of energy, and generally lipids are insoluble in water. So the problem of transportation in the aqueous plasma is solved by associating non-polar lipid. So how this lipid is basically transported in the plasma is by associating this lipid, which is non-polar, and main component of lipid is triacylglycerol and cholesterol ester. So these lipids are associated with proteins basically. So these lipids, along with the phospholipid and cholesterol and protein, these all are associated together. It forms a structure. The structure is known as lipoprotein. That's why the name lipoprotein. It consists of lipid component and protein component. So lipoprotein consists of a non-polar core. So core means the inside of this structure, a spherical structure. It will have more non-polar substances, which will include what? Lipids like cholesterol ester and cholesterol ester and triacylglycerol. And the surface layer will be made up of amphipathic lipids. What are the amphipathic lipids? It is generally cholesterol and phospholipids. And this surface will contain different types of proteins also. So the non-polar lipid core consists of mainly triacylglycerol and cholesterol ester. Okay, so lipoprotein structure here we are describing. Okay, so lipoprotein consists of non-polar core and surface layer of amphipathic lipid. And what non-polar core is consisting of? It is mainly consisting of triacylglycerol and cholesterol ester, and it is surrounded by a single surface layer of amphipathic phospholipids and cholesterol. So see what all components are there in the structure of lipoprotein. The core of the lipoprotein is non-polar, which is mainly made up of triacylglycerol and cholesterol ester. The surface is mainly made up of amphipathic lipid. And what are those amphipathic lipid? It is phospholipid and cholesterol. These are oriented so that their polar group face outward to the aqueous medium. Of course, that's the point of forming this lipoprotein, to carry the lipids, mainly which lipids? Mainly triacylglycerol and cholesterol ester. To carry triacylglycerol and cholesterol ester in the aqueous plasma, this structure is formed. So their polar end will be facing outward. And the protein moiety of a lipoprotein is known as apolipoprotein or apoprotein. So this apoprotein will be embedded in the surface layer of amphipathic lipid. So this is a structure you see. Inside, this is the core. Here it is present triacylglycerol and cholesterol ester. You can see it's very clearly written. This is the core. It is made up of mainly triacylglycerol and cholesterol ester. And cholesterol ester and triacylglycerol are generally non-polar. And this surface layer is made up of amphipathic lipid, which consists mainly of phospholipid and free cholesterol. It looks like any biological membrane, right? So it is made up of phospholipid and free cholesterol. And this surface layer will contain different proteins. Those proteins will be known as apoprotein. So this can be peripheral apoprotein or integral apoprotein. And the proteins generally, which is the part of lipoprotein, is named as ApoC. Like that name will be there: ApoA, ApoB, ApoC, ApoE, like that. So this is ApoC and this is ApoB. So it's very clear. You see, the core is made up of triacylglycerol and cholesterol ester. Okay, simple. The surface layer is made up of phospholipids and cholesterol and phospholipid, which is amphipathic. The core is non-polar, and the surface layer of amphipathic lipid contains proteins known as apoprotein or apolipoprotein. Examples are ApoA, ApoB, ApoC, like that. So some apoproteins are integral and which cannot be removed, where others are freely transported to other lipoprotein. The apoproteins which are not integral apoproteins, which are basically the surface apoprotein, peripheral apoprotein, can be exchanged between different different lipoproteins. So this is the general structure of lipoprotein. Triacylglycerol cholesterol ester core. Surface is made up of phospholipids and cholesterol. Surface contains apoproteins. Examples are ApoA, ApoB, ApoC. Now the classification of lipoprotein. This is important. This might come in your exam also. And before we proceed, like I will tell you all to find, like, have the previous year's question, 5 year or 10 year whatever, and for whatever topic generally we teach, you have to go read from the book also and from notes whatever you are making, and also you have to see the questions like previous 5-10 years question, that if some topic which we are taking, something else they have asked apart from that, so you should tell us. Suppose I am taking lipid, right? Lipid chemistry and metabolism. So most probably next or next to next class will be the last class. So you tell me by next class these topics are left from the lipid, which has come in exam but I have not covered, so I'll try to cover that. Okay everyone.
Okay, sir.
Okay, so now the classification of lipoproteins. So this can be classified in various ways. So first we will see that in the classification of lipoprotein based on the density. Okay, so these lipoproteins contains lipids and it contains some protein also. So because it contains lipid, it will have, depending on the lipid content, it will have different density, right? So this can be all the lipoprotein mixture can be separated with the help of method known as ultracentrifugation, and depending on the sedimentation coefficient or flotation coefficient, SF, 5 major groups of lipoprotein have been identified. Okay, so in body generally 5 groups of lipoprotein is identified, and all those 5 groups have different structure, different composition, different role, clinical significance. So why the clinical significance is different? Because of the different content of the lipids and proteins in them. First group of lipoprotein under the classification which is based on density is chylomicron. So from where this chylomicron comes? Sorry, this is a typo. Chylomicron. This is RON. So this chylomicron are generally derived from the intestinal absorption of triacylglycerol and other lipid. So this chylomicron will form generally in the intestine. So its main constituent will be the absorbed, whatever we are eating, the absorbed triacylglycerol and other lipid, that will be the main content of the chylomicron. So if someone asks where chylomicron is formed and what is its main content, so chylomicron is formed in intestine and its main content is triacylglycerol and different lipids what we eat, and its density is the lowest basically, because it has higher lipid content. It is less than 0.95, while the mean diameter is more. So it will be this chylomicron will be the bigger, we will see also relatively will be bigger and less dense. Second group, first was chylomicron based on the density. Second group of lipoprotein based on density is very low density lipoprotein. So chylomicron, we saw that it is formed in the intestine and derived from the mainly the lipids we eat. And a very low density lipoprotein VLDL, this is mainly derived from the liver. And this is why it is formed. It is formed from the liver, which contains triacylglycerol and other lipid, so that it can be exported to different organs. So VLDL mainly consists of triacylglycerol and this is formed in liver, and its density is higher than the chylomicron. Even if you don't know exactly, this is fine, but it is higher than the chylomicron and its diameter is smaller than the chylomicron. Third is intermediate density lipoprotein, ideal. So this will be formed from the metabolism. It means by the breakdown. So the breakdown metabolism is known as catabolism. So ideal is formed from the breakdown of VLDL, whose density will be between VLDL and LDL. So the density of ideal will be more than the VLDL, less than the LDL, and its diameter will be smaller than the VLDL. So chylomicron is formed in the intestine, VLDL is formed in liver, and then it is coming out of the liver for the export of triacylglycerol to different tissues, and once it is in the circulation, this VLDL is converted to IDL, okay, intermediate density lipoprotein, and then IDL will be converted to LDL. This LDL represents the final stage of catabolism. I told from VLDL IDL form, from IDL LDL form. So this is the final stage of catabolism of VLDL, and its density is higher than the VLDL and IDL both, and its diameter is smaller than the VLDL and IDL. So you understood from where chylomicron is coming, from where VLDL is coming, and how IDL and LDL is forming from the VLDL. So these all are classification based on the density because of the different densities and different sizes. Then next is high density lipoprotein. It is mainly involved in the reverse cholesterol transport. What is the meaning of reverse cholesterol transport? It means it carries cholesterol, it transports the cholesterol from different organs and different tissues of the body back to the liver. So it is opposite of the VLDL and LDL. It involves cholesterol transport, and also in VLDL and chylomicron metabolism, we will see how in VLDL and chylomicron metabolism HDL is required because it gives the apoproteins. The apoproteins required for the formation of VLDL and chylomicron comes from the HDL. We will see when we read about the HDL metabolism, and its density is the highest of all the lipoprotein based on density, okay, and its diameter is the smallest. So here you see this is chylomicron, the biggest, then VLDL is smaller than that. Here itself you see like revise where chylomicron comes. Chylomicron comes from the intestine, and it mainly consists of the triacylglycerol and lipids what we eat. VLDL comes from liver, and it is mainly consisting of triacylglycerol, which is formed or stored in the liver, and then this triacylglycerol is basically exported into the different organs of the body from the liver to all the tissues, okay. So this is the for that, and then this VLDL will form IDL and LDL subsequently in the circulation, and this HDL has opposite role. It will carry the cholesterol from different organs of the body back to the liver, okay. And this is required for the metabolism of VLDL, HDL, IDL. How? And chylomicron. How? Because it gives this HDL gives the apoproteins required for the formation of maturation of the VLDL and chylomicron also. And here you can see that this is the total lipid content, the top, and this is the protein content. So as the molecule becomes smaller in the classification from chylomicron to HDL, its protein content increases and lipid content decreases. Lipid content is 99, 92, 85, 80 and 50, and its protein content is 1, 8, 15, 20, 50. Okay, so in classification, when comes classify lipoprotein, then you have to classify according to density. That is one type. Then you have to write where proteins are more and where lipids are more. Okay. So now the next classification based on electrophoretic mobility. So that was based on ultracentrifugation. Once you do centrifugation of the lipoprotein mixture, that will get separated based on the density into chylomicron, VLDL, LDL, IDL and HDL. So that was the classification based on density by ultracentrifugation. Now because this lipoprotein contains protein also, so they will be separate, that can be separated with the help of electrophoresis also, right? So once you separate according to electrophoresis, it will form different band, and different bands in the electrophoresis will contain different lipoproteins. So this lipoprotein may be separated according to the electrophoretic properties into alpha, pre-beta, beta and broad beta lipoprotein. So the names according to density is chylomicron, VLDL, low density, very low density. So you can understand that from the name itself that on what basis the classification has been done. And when someone tells alpha lipoprotein, beta lipoprotein, pre-beta lipoprotein or broad beta lipoprotein, it means they are classifying this lipoprotein based on the electrophoresis. Okay. The mobility of lipoprotein is mainly dependent on the protein content, right? Because the protein part of the lipoprotein contains charge, and if we give what is electrophoresis, basically when you give the electric supply, basically in electrical field, the charged molecule, so these proteins are charged molecule, start moving, and it will move at the different rate based on its charge, its size and all those things, and then it will separate into alpha, beta, pre-beta and all like that. those with higher protein content will move faster towards the anode, right? So it will move faster towards the positive electrode because the protein normally has negative charges. At the pH we do electrophoresis, all the proteins generally contains negative charge. So whichever protein will have high, whichever lipoprotein will have higher protein content, it will move faster towards the positive electrode, towards anode, and those with minimum protein content will have minimum mobility. So it will move slower if protein is less. So this is like electrophoretic band, electrophoresis slide. Okay, so suppose if you are putting here, it will start moving like this. So once it moves, and then with the help of a densitometer, it can be made into a graph. Okay, so lipoprotein may be separated according to their electrophoretic property into alpha, pre beta, beta, and broad beta. So what are those actually, these alpha and beta, pre beta, which it is corresponding to? So basically HDL are alpha. Alpha lipoprotein, if someone says, that is equivalent to HDL, okay, high density lipoprotein. Then VLDL is pre beta, and beta is LDL, and IDL are broad beta. And free fatty acid and albumin complex, although they are not lipoprotein, but they are also lipid fraction in serum. So when you do centrifugation, the content of the albumin and free fatty acid will also form a band, and chylomicron will remain as at the origin, because we saw that it is mostly lipid. Here the protein content is very low. If you go back and see here protein content is 1, so it has almost no protein content. So when you do electrophoresis, it will remain at the origin site, it will not move much. But as the protein content increases, it will move. So which will move the furthest? HDL, because it has the highest protein content. Then LDL, IDL, and VLDL. But one thing, if we read here, we'll see that VLDL has less protein content, right? You see, VLDL has less protein content, so ideally it should be behind, it should form band after LDL. First band should be made up of HDL, because it has the highest protein content, so it will move furthest. Then LDL, then VLDL should come, and then chylomicron. But this VLDL transferred faster than the LDL, because even though its protein content is low, the type of lipoprotein which is present, that has faster mobility. Okay. So VLDL with less protein content than LDL move faster than LDL, even though LDL should move faster because it has more protein content. But this anomaly is because of the nature of lipoprotein present in VLDL, that has the higher mobility even if the content is low. So just know that according to main classification is according to ultracentrifugation based on density. Another classification is based on the electrophoretic mobility. In electrophoretic mobility, the bands will be alpha, pre beta, beta, and broad beta. So alpha corresponds to HDL, pre beta can correspond to VLDL. That's the name, like it is coming before beta. Ideally it should be alpha, beta, and then VLDL, but it comes before LDL, VLDL. That's why it is named as pre beta. And then ideal are broad beta lipoproteins. Chylomicron stays at the origin point only, wherever you are keeping the sample. It will not move because it has almost no protein. So this is the summary of the classification: HDL, LDL, IDL, VLDL, chylomicron. Here you see the protein content. Protein content, HDL has highest, then LDL, then IDL, then VLDL, then chylomicron. Then combining lipid and triacylglycerol content, if you see, this is the main lipid non-polar content. You see, chylomicron has so much triacylglycerol, VLDL has less than that, then IDL, then LDL, HDL, like that. And size also, you can see density, you will see the highest, HDL has highest density, and then chylomicron has lowest density because it has highest fat content, and diameter is also corresponding to the relation with the density only. So now classification of lipoprotein based on the nature of apoprotein, the protein content. So different types of lipoprotein, as we read, what are the major types of lipoprotein? HDL, LDL, VLDL, IDL, and chylomicron. So generally all these lipoprotein will have many different types of apoproteins also. So one or more apoproteins or apolipoprotein, which is basically protein or peptides, are present in each lipoprotein. So the major lipoprotein of LDL is alpha lipoprotein according to electrophoresis, are designated A. So ApoA is mainly the major lipoprotein of HDL. So remember that ApoA is mainly associated with HDL, and the LDL, the major protein is ApoB, apolipoprotein B. LDL and VLDL both contains LDL and VLDL both contains ApoB. HDL contains ApoA lipoprotein, ApoA apolipoprotein, and LDL and VLDL contains ApoB. And here you see what is B100. Okay, chylomicron also contains ApoB, but it is B48. We'll see how it happens. Now LDL and VLDL, the ApoB, which is the apolipoprotein part of the lipoprotein, is produced in the liver, because LDL is formed in the, we saw that VLDL is formed in the liver. Now chylomicron is formed in the intestine. So the protein also, the protein part, which is the ApoB B48 part of the chylomicrons, also will be formed in the intestine. So ApoB48 is present, synthesized in intestine, and ApoB100 is synthesized in liver. And ApoE is found in most of the basically lipoproteins, but not in LDL. ApoE is found in VLDL, HDL, chylomicron, and chylomicron remnant. So the functions of apoproteins. So in exam classification and function, then you have to write like this. Classification mainly based on the density, and then alpha beta you can write also. So they can form part of the, we saw that apoprotein are what the function of apoprotein. It is mainly, it forms the structural part of the lipoproteins. It is combining with the surface amphipathic lipids, right? So ApoB, a structural component of VLDL and chylomicron. ApoA, the structural component of HDL. Now apart from its structural function, apoprotein has some enzymatic activity also. So these apoprotein also act as enzyme or enzyme cofactor. So this C2, ApoC2 is the cofactor for lipoprotein lipase. Lipoprotein lipase helps in the breakdown of triacylglycerol. Okay, this I had told in the lipid chemistry class only. So there the cofactor required is ApoC2. Now ApoA1 is required as the cofactor for, enzyme cofactor for lecithin cholesterol acyl transferase. So this helps in the formation of cholesterol ester. So LCAT, this is the enzyme, lecithin cholesterol acyl transferase. This is the LCAT enzyme. It requires enzyme cofactor ApoA1. Either it can act as enzyme cofactor, or it can act as enzyme inhibitor also. So A1 is the activator of LCAT, and ApoA2 and C3 is the inhibitor of lipoprotein lipase. What is the activator of lipoprotein lipase? ApoC2. What is the inhibitor of lipoprotein lipase? ApoA2 and ApoC3. And ApoC1 is the inhibitor of cholesterol ester transfer protein. So even if you don't remember this much, first function you should definitely remember is that this is the structural component of lipoproteins. Then you can give example, ApoA is the structural component of HDL, ApoB is the structural component of VLDL and chylomicron. Then second, even if you don't remember the example, but definitely write that it acts as the enzyme cofactor and enzyme inhibitor also. And if you remember the example, then good only. Third is they can act as the ligand for interaction with lipoprotein. So like in that, like tissue receptors present in the body, right, in the liver and the other organ, for these lipoproteins to bind. So these ApoB100 and ApoE, which is present on LDL, and ApoA1, which is present on the HDL, mainly they act as the ligand also. These part of the lipoprotein bind to the lipoprotein receptor, and that way it helps in the binding of the lipoproteins. So it acts as ligand also for binding with the lipoprotein receptor. So first a structural component, second enzyme cofactor and inhibitor, third ligand for binding with the lipoprotein receptors. Now metabolism of chylomicron. So we have to see the metabolism of chylomicron, LDL and HDL. Today we will see for chylomicron only, and then other two we will see in the next class. So metabolism of chylomicron. So these chylomicron are found in chyle, formed only from the lymphatic system, draining the intestine. So basically, definitely, because it is forming, chylomicron is forming in the intestine, so it will come out with the help of chyle into the lymphatic system, and then from lymphatic system it will come into the main circulation, and they are responsible for the transport of all dietary lipids, mainly TAG, triacylglycerol, into the circulation. So how this chylomicron is synthesized? First, we'll see the component. What is the component? Very important component of chylomicron. We saw that apoprotein, ApoB, and especially ApoB48. So this ApoB48, this chylomicron contains ApoB48, which is synthesized in the rough endoplasmic reticulum. Okay, and How this is formed? This is formed because of the RNA editing. So for any protein, what is present? DNA is present, right? Gene, and then the transcription happen, and then mRNA is formed, and from mRNA protein will be formed. So for this also, for because this is ApoB48 is a protein, which is a protein, so it means there must be some mRNA for this. So that mRNA is formed from the gene. So but that mRNA, the same mRNA codes for ApoB48 and ApoB100. Both the proteins are coded by the same mRNA. But in ApoB100, the full mRNA is translated. Translation is basically what? From mRNA to formation of the protein. So when ApoB100 is formed, the full mRNA is translated to ApoB100. But that happens in the liver. But in the intestine, what happens? That this mRNA, which is coding for the ApoB100, some editing happens at the 48. Once 48 amino acids has added, 48% of the amino acids, editing of the mRNA happens and the translation stops because of the insertion of stop codon. So then it is full B100 is not forming, only 48 is forming. See here. So this is just understand that this says some gene, okay, the gene for ApoB, right? Just understand one by one. So there is gene which is present for ApoB, that is present in intestine also, liver also. In liver, that transcription will happen, and intestine, both the transcription will happen. So mRNA will form in the liver also and in the intestine also. But here what happens? This mRNA, when translation is happening, this full mRNA will be translated into the apoprotein, which will be known as B100. It means the total amino acid, you see the total amino acid present in ApoB100 is 4563 amino acids will get coded, and it will form the protein. But when the same translation is happening in the intestine, this mRNA, instead of CAA, UAA gets added in the during the protein formation, during the translation. So instead of formation of the complete ApoB protein, which would have contained 4563 amino acid, the translation stops at the 48% level. So then it will contain the only 2152 amino acids only. So that's why the name ApoB48. It means only 48% of the translation completed and stop codon came. So once stop codon come, no further translation can proceed, right? So this is like truncated. This is the smaller version of ApoB100 which is forming in the liver. But the gene and mRNA for both ApoB100 and ApoB48 is same. So once you see this slide one or two times, you will be able to understand how from the same gene and from the same mRNA, two different length of the protein is forming. So that's why the synthesis of ApoB48 is the result of RNA editing. Okay. Now we saw that one very important component of chylomicron has formed, ApoB48, right? So in liver, the single ApoB, this is what they are describing again. So the single ApoB gene is transcribed into an mRNA. This is simple. If you read, you will understand this. That directs the synthesis of 100 kilodalton protein, ApoB100. In the intestine, the same gene directs the synthesis of primary transcript. Primary transcript is basically mRNA. So same gene is forming the same mRNA. However, one enzyme which is cytidine deamine, if you don't remember, it's fine, converts CAA codon to UAA codon, which is a stop codon. So rather than forming glutamine, CAA codes for glutamine, we saw GLN. So CA would have coded for glutamine, and then further coding of protein would have proceeded from the mRNA. It codes for UA, which is the stop codon. So that's why the translation stops at the 48 kilodalton protein level. So once, even if gene is same, mRNA is same, if different protein is forming, it will have different function in the two organs. So that's why ApoB100 helps in the formation of VLDL and LDL. ApoB48 helps in the formation of chylomicron. So where it is produced, this I told in rough endoplasmic reticulum, right? So apolipoprotein B is synthesized in rough endoplasmic reticulum, here, like this rough endoplasmic reticulum, and it is incorporated into the lipoprotein in the smooth endoplasmic reticulum. So once this apoprotein is formed, this will come and combine with the lipoprotein in smooth endoplasmic reticulum, which is the main site of synthesis of triacylglycerol, and triacylglycerol is the main component of the core of the lipoprotein. So basically triacylglycerol, and it is containing the amphipathic lipid in the smooth endoplasmic reticulum that combines with the apolipoprotein B40H, which is coming from the rough endoplasmic reticulum, and then it will contain the additional carbohydrate residue in the Golgi apparatus, and then this chylomicron, when it is formed, will pass into the lymphatic channel. This is the blood capillary and this is the lymph vessel. So this will pass into the lymphatic channel and it will come into the lymphatic system, and from lymphatic system it will come into the main circulation. So what happens in A beta lipoproteinemia, which is a rare disease? A beta lipoproteinemia is it means beta lipoprotein is deficient. Lipoprotein containing ApoB are not formed and lipid droplets accumulate in the intestine and liver due to non-formation of VLDL. So when in A beta lipoproteinemia, lipid contains basically all the lipids, it cannot come out in circulation, because for lipid to come out in circulation it requires chylomicron from the intestine and VLDL from the liver. So VLDL and chylomicron maturation will not happen because of the absence of beta lipoprotein. So all the lipids will get accumulated in the intestine and liver. Now first was apolipoprotein content of the chylomicron. Now synthesis of lipid and formation of lipoprotein of the chylomicron. See, so long chain fatty acid are esterified to yield triacylglycerol. So whatever we are eating, either we are eating directly triacylglycerol or fatty acids, those will also get esterified and form triacylglycerol in the mucosal cells of the intestine. So, and together with the other products of lipid digestion are incorporated into lipoprotein in the smooth endoplasmic reticulum. So in smooth endoplasmic reticulum, triacylglycerol and other lipids, whatever we are eating, that will combine together and form lipoprotein, core lipoprotein content of the chylomicron in the smooth endoplasmic reticulum, and the ApoB will come from the rough endoplasmic reticulum, and then carbohydrate residue will be added in the Golgi apparatus, and then this chylomicron will be released into the from the mucosal cells into the lymphatic channel by reverse pinocytosis. Okay, with the help of pinocytosis, normally endocytosis happen, here exocytosis is happening basically. And then this chylomicron will pass into the lymphatic system and it will eventually enter the systemic circulation. And once it is in systemic circulation, remember this, once it is in systemic circulation, chylomicron will be acted upon by lipoprotein lipase. So what will lipoprotein lipase do? Basically lipoprotein lipase will basically break down the triacylglycerol. So reaction with lipoprotein lipase result in the loss of approximately 90% of triacylglycerol of chylomicron. So lipoprotein lipase breaks down triacylglycerol. So that will result in loss of approximately 90%. So most of the triacylglycerol content of the lipoprotein will be broken down by the lipoprotein lipase in the circulation. And then the ApoC, which is present in the chylomicron, will also be lost and it will be returned to the HDL, but not ApoE. ApoE given by the HDL remains with the chylomicron. And now this chylomicron, which is in circulation, which has now lower ApoC and triacylglycerol content because of the activity of lipoprotein lipase, is known as chylomicron remnant, because this is not now full chylomicron remnant, right? This is not full chylomicron because this has very less triacylglycerol, because most of the triacylglycerol got lost because of the activity of lipoprotein lipase in the circulation. So this chylomicron remnant, which is much smaller, half the diameter of the parent chylomicron, and now because triacylglycerol is reduced but cholesterol and cholesterol ester is not reduced, so it will have relatively more cholesterol and cholesterol ester. And this chylomicron remnant, which has less TAG and more cholesterol and cholesterol ester, will be taken up by liver with the help of receptor mediated endocytosis, and cholesterol ester and triacylglycerol are hydrolyzed and metabolized. So once it enters the liver, whatever little bit of triacylglycerol and cholesterol ester present in the chylomicron remnant will be hydrolyzed and metabolized. Okay, and this uptake of chylomicron remnant into the liver is mediated by ApoE, because we saw in the previous slide that with the help of lipoprotein lipase, triacylglycerol and ApoC is degraded but not ApoE. ApoE remains with the chylomicron remnant. Now the hepatic lipase, there are two types of lipase: lipoprotein lipase, which is present in the circulation. In the circulation, lipoprotein lipase breaks down the triacylglycerol carried by lipoprotein. Now hepatic lipase, the lipoprotein lipase which is present in the liver, it has two roles. It acts as a ligand to facilitate remnant active. It means what? This lipoprotein lipase which is present in the liver, which is known as hepatic lipase, it helps in the taking up of chylomicron inside the liver, and it hydrolyzes remnant triacylglycerol and phospholipid. So whatever the remaining triacylglycerol and phospholipid present in the chylomicron remnant will be broken down by the lipoprotein lipase present in liver. And the products which are released by the metabolism of chylomicron remnant are reutilized for the synthesis of VLDL, because I told you already VLDL is formed in liver, chylomicron is formed in intestine. So whatever chylomicron remnant part goes inside the liver after its metabolism, those products of the chylomicron remnant is used by the liver to form VLDL. So this is the last slide. You see the how chylomicron is getting broken down. Chylomicron formation you saw well, right? How ApoB48 is forming. Then how in a smooth endoplasmic reticulum we saw that triacylglycerol and lipids will combine and form the core of the chylomicron. Then ApoB48 will add from the rough endoplasmic reticulum. Then carbohydrate residue will be added in Golgi apparatus. And then this whole chylomicron will come out with the help of reverse pinocytosis into the lymphatic channel, and from lymphatic channel to systemic circulation. Once in systemic circulation it comes, it will be acted upon by the lipoprotein lipase which is present in the circulation. Okay, so this lipoprotein lipase will mainly break down most of the triacylglycerol content of the, and we know that the major content of chylomicron is triacylglycerol. So these triacylglycerol will be broken down, almost 90% will be broken down by the lipoprotein lipase present in the circulation, and ApoC also will be degraded. And but ApoE and the little bit of cholesterol and cholestyl ester and little bit of triacylglycerol will form the part which will be known as chylomicron remnant, and this chylomicron remnant will be taken up by the receptor and into the liver, and this receptor taking up is helped by the ApoE. Remember, ApoE helps in the taking up of chylomicron remnant into the liver, and hepatic lipase also. So lipoprotein lipase two types, one is present in circulation and another is inside the liver. So the hepatic lipase also helps in the taking up of chylomicron remnant. So once the chylomicron remnant goes inside the liver, it will be acted upon by the hepatic lipase, lipoprotein lipase, and it will break down the remaining triacylglycerol and phospholipids, and the product formed from this metabolism of chylomicron inside the liver will be taken up for the formation of VLDL. And the metabolism of VLDL, LDL and HDL we will see in the next class. So thank you.
Thank you, sir.
Bye bye, sir. Thank you.
Okay, bye.